In this lecture I will talk about T-cell activations, how T-cell get activated. But in the beginning I want to mention you something about the T-cells. So T-cell is actually broadly categorized into two subsets. One is called T helper cell and another is cytotoxic T cells, depending on the co-receptor. This T helper cell express CD4 co-receptor and the cytotoxic cell express CD8 co-receptor. Okay, now these two cells can recognize two different MHC molecules. This cytotoxic T cells express MHC1 and these cells actually recognize MHC2. So those cells express class 2 MHC molecule can activate T helper cell, and those cells express class 1 MHC molecule can activate cytotoxic T cells. Okay, now depending on the activation status, whether it is activated, the cells are activated or not, they can be divided into both two cells can be divided into three categories. One is called knife cell, another is called effector, and last is memory. What happens when these TH cell or cytotoxic T cells, TC cells, are actually inactive, or you can say when these cells, TC cells or T helper cells, have not yet recognized any pathogen or antigen, it is called knife cells because it is inactive. Okay, when this knife cells get activated, then it becomes effector cells, and after the elimination of the pathogen or the antigen, some effector cells survive. Many effector cells, many of them die, but some effector cells survive, and that effector cells become memory cells. And I want to mention here one important cells called professional antigen presenting cells. So these cells are actually very professional to present antigen. Okay, these cells are actually three. There are three cells that is actually called professional antigen presenting cells, like dendritic cell, macrophage and B cells. These three cells are called professional antigen presenting cells because they are very professional. This dendritic cell can activate knife cell, effector cell and memory cell, but this macrophage cannot activate knife cell, can activate effector cell and memory cell. Also B cell cannot activate knife cell but effector cell and memory cell, because in this class I am going to talk about how dendritic cell actually activate the inactive T cells, that is called knife T cells, because after activations by the help of dendritic cells, these cells, effector cell and memory cell, can be activated by this macrophage and B cells. What happens actually, these macrophage and B cells also get help from the helper cells. Okay, but the knife cell, activation of the knife cell is actually solely responsible for this help of these dendritic cells. So dendritic cells actually activate knife cells. So in this class I will talk about how dendritic cells activate knife T cells. So what happens in the tissue site, you can also call it organ, tissue or organ, there is already some dendritic cells are present. Some dendritic cells are already present, but this dendritic cell is in inactive conditions. Inactive. Let's assume there is infections. Some pathogens are present in the tissue. So when the tissue or the organ get infected by any pathogen, it can be virus, bacteria, fungi, protozoa, helminth. When this pathogen infect the tissue, there is already some dendritic cell, macrophage are present. These are called sentinels. They can sense this pathogen by the help of a receptor that can recognize the pathogen. These are called pattern recognition receptors. I have already discussed about what is pattern recognition receptor in my previous class. The example of pattern recognition receptors are toll-like receptor, that there are nod-like receptor, there are C-type lectin receptor, retinoic acid-like receptor. So there are different pattern recognition receptors, like toll-like receptor can be 1 to 9. So there are different type of pattern recognition receptors are already present in inactive dendritic cells that actually help to sense the pathogen. So when there is no infection, the dendritic cells remain as inactive, but if there is any infection by any pathogen, the dendritic cell can sense that there is some infection, and these dendritic cells become activated. It becomes activated. So activated dendritic cells. Now some activated dendritic cells leave the infected tissue site. These are the infected tissue site. Infected. This dendritic cell, some dendritic cell leaves the infected tissue site into this activated dendritic cells into nearest lymph node or lymph gland, you can call, where TC cells or knife T helper cells, knife in conditions that the police are actually patrolling the police stations. So there are knife T helper cells are patrolling, knife cytotoxic T cells are patrolling in each and every lymph node or gland in our body, and when dendritic cells, activated dendritic cells, reach into the lymph node, they activate these T helper cells. So they activate. So before the activation of knife TH cell or knife TC cells, the dendritic cell must actually get activated. At first it must be activated. Okay. Another route of infection or pathogen can enter, that is blood. Through blood a pathogen or antigen can enter, and when pathogen or antigen enter through the blood, it do not go to the lymph node. The antigen or pathogen go to the spleen, another secondary lymphoid organ, spleen, and in the spleen there is already dendritic cell are present that is inactive in conditions. Dendritic cell are present that is inactive. Now same thing occur. The dendritic cell express pattern recognition receptors. They recognize the pathogen, gets activated, they get activated, and in the spleen this T helper cell, knife T helper cell and knife cytotoxic cell also patrolling, they can actually activate it by these dendritic cells. So there are two routes through which pathogen or antigen can enter. Now let me draw the interactions that occur between this dendritic cell and the T helper cell and cytotoxic cells to activate it, to get activated. Let's assume this is a dendritic cells. Let's assume this is a dendritic cells. So it is activated. This activated dendritic cell can express both class 1 and class 2 MHC molecule. Why? Because activated dendritic cell is a professional antigen-presenting cells, and a professional antigen-presenting cells can express MHC-2 molecule, can express. And also, as the dendritic cell has nucleus, as it has a nucleus, it also expresses MHC-1 molecule. Okay. Now what happens when these dendritic cells actually present in the tissue or when it is present in the spleen, they actually engulf or phagocytose the pathogens and chop it, process the antigen and present the peptide fragment of the antigen by the help of MHC-1 and MHC-2 molecule. And let's assume this is knife T helper cell and there is knife cytotoxic T cells. Okay, these cells can express T cell receptor. So these are the T cell receptor by which they recognize the MHC and peptide complex. So let me draw here. Let's assume this is from the dendritic cell, the molecule that is expressed by dendritic cell and molecule that is expressed by T cells. So first interactions that occur in between dendritic cell and the T cells is, the first thing is MHC-1 or 2 with peptide complex, peptide, interact with T cell receptor. At first, this is the first signal, so I can write it, the first signaling or first interaction that helps to activate T cells. Okay, so what happens? There are many cytotoxic T cells and T helper cells. There are many T cells and T helper cells, but among them very few cells are present that can recognize a particular antigen. What happens if I have 10 million TC cells or T helper cells, then I have 10 million different type of T cell receptor. Why? Because due to the VGJ recombinations, diverse set of T cell receptor are produced during the development of T cells, and if I have 10 million T cells, then I have 10 million type of T cell receptor, and this 10 million type of T cell receptor that is unique can recognize 10 million different antigen. So there are many different T cells present, they expressing unique T cell receptor. Among them, very few can recognize this type of antigen and they are actually selected. So this process is called clonal selections. This is very important. Now second interaction occur, that is this naive cell express, this naive TC cells and these cells express a receptor that is called CD28. This receptor is called co-stimulatory receptor, and this activated dendritic cells, activated, when the dendritic cell is inactive, it do not express this molecule, but when this dendritic cell get activated, it starts to express this molecule that is called CD80 or CD86. This is co-stimulator molecule. This is also known as B71. You can write it B71 or B72. This 80 is called B71 and 86 is called B72. Do you know what happens if only these T cells interact with this MHC molecule, not with the CD80 or CD86? They become anergic. So if there is no recognition between the CD28 and CD86 or 86, then it becomes anergic. Anergic cell means unresponsiveness or non-functional cells. So only activated dendritic cell can activate a naive cell, naive T cells, because when the dendritic cell is inactive in conditions, it do not express CD80 or 86, and when this CD80 or 86 do not actually engage with this CD28 molecule, the cells become anergic. So if there is no interactions, then one thing occurs that is called anergy. That means the cells become non-functional, it cannot function. What happens? When the dendritic cells is not infected or activated, it can represent antigen or peptide fragments by the class 1 and class 2 MHC molecule, because this is called turnover. All proteins get turnover after its half-life. So its cell own protein can be presented by this MHC1 and MHC2 molecule. And there are some self-reactive T cells that can recognize the self-antigen. But when this dendritic cell is actually presenting self-antigen, it is not activated in conditions. So they do not express CD80 and 86. So self-reactive T cells that recognize T cell receptor and this self-antigen, but without the interacting with the CD80, 86 become anergic. So this is actually peripheral tolerance. This is called peripheral tolerance. I have discussed about this in my previous video. There are two types of tolerance occur. That is central tolerance that occurs in the primary lymphoid organ, and a secondary or peripheral tolerance that occurs in the secondary lymphoid organ like spleen or lymph node. This is the second interactions. For the proper activation of T cells, this interaction is very important. So if this interaction do not occur, the cells become anergic. It is for self-reactive T cells. So anergy occurs only for self-reactive T cells, not with normal cells. So now I can write the second interactions, that is between the CD80 and CD86. Both molecules can interact with a receptor present in the T cell, that is CD28. This is called co-stimulations, because this molecule are called co-stimulator molecule, and this is co-stimulator receptor. Now when this second interaction occur between the dendritic cell and the TC and naive T helper cells, two major important events occur. I want to mention here. The first event for these interactions, two major important events occur. First is expression of a molecule called CD25 and a molecule called CD40L by T cells. Both these molecules is expressed by T cells. Let me draw. Let's assume this is a CD25. Both these cells can express CD25, but I have to here mention it, the CD25 in more details. What is CD25? So let's assume this is a naive T helper cell or cytotoxic T cells. Okay, when this cells is naive in condition or inactive in conditions, they express a receptor that is called IL-2 interleukin 2. It is a cytokine receptor. This interleukin 2 receptor actually have 3 subunits, but when the T cells is naive in condition, they express only the 2 receptor, only 2 subunit of this receptor, that is called beta and gamma. Okay, but when this naive T helper cell or cytotoxic T cells engage with T cell receptor MHC and CD28 and CD86, then it starts to produce by itself another subunit of this IL-2 receptor, that is called alpha segment or alpha subunit. So this is called CD25. So I can write that CD25 is equals to IL-2 receptor, that is alpha subunit. Okay, here is very important role of this CD25. This is CD25. Very important role of the CD25. What? It has been seen that this beta and gamma is constitutively expressed by these naive cells, but when this CD25 molecule it starts to express in the activating naive cells, the affinity towards IL-2 cytokine, because this molecule or this is actually receptor that can bind with IL-2 cytokine, the affinity towards the IL-2 cytokine increase approximately 100 folds. So now you can understand this CD25 actually increase the affinity towards the IL-2 cytokine. And what is IL-2? I have to explain here. IL-2 is actually a cytokine that is expressed by these cells. They are actually producing IL-2. This is IL-2. They are starts to express many CD25 and they are expressing many IL-2 cytokine. And this fashion of signaling is called autocrine signal, autocrine, because the cells is producing its own cytokine and receiving the cytokine. So this CD25 actually help in the increase of the affinity of this IL-2 cytokine, and the cells actually starts to produce IL-2. And the role of this IL-2 is the IL-2 secretions actually perform a very important role. You must remember, I am writing here, that is called clonal expansion. What I have told, that there are many T cells, TC cells and T helper cells, but among them very few can recognize this type of antigen, because each and every T cell receptor is unique. Now to increase its number or expand, it must go through a expansion process or proliferation by the help of IL-2. So IL-2 cytokine actually helps to proliferate. This is called clonal expansion. So at first clonal selection occur. This is clonal selection. The cells are selected and now they are expanded. This is very important. By the help of IL-2 signaling, the cells are producing itself IL-2 and it gets proliferated. So this is clonal expansion occur. So these interactions CD80 and 86 and CD28 is very important, because for this interaction CD25 starts to express and IL-2 actually bind with this receptor with 100 times increase in affinity. So these cells gets proliferated. Okay, another thing occurs, that is called CD40L. What is CD40L? But this molecule is only expressed by CD4 cells, T helper cells, not the cytotoxic T cells. So this signaling do not occur in this case, but in this case, in this cell. What happens? The cell starts to express a ligand that is called CD40L in response to this second signal. So for this second signal, this is occur. CD40L expression starts to, these cells express CD40L, and the activated dendritic cells express a receptor for the CD40L, that is called CD40. So now I can write that after this, what happens? This T cells actually starts to produce CD25 and they actually bind with IL-2, and for this reason they actually go clonal expansion. And another thing occurs, that is expression of CD40 ligand and receptor CD40 by these dendritic cells. When the CD40 and CD40L ligand bind with each other, they help to increase the expression of, when these two molecules bind with each other, another thing occurs, that is this activated dendritic cells starts to express CD80 and CD86 molecule more. So these activated T cells are expressing CD80, 86, but when the CD40 and CD40L engage, this dendritic cells starts to express more CD80 and 86, so that more CD28 can bind with each other. So more signaling it will get, so more it will produce CD25, and a autocrine signaling occurs, and it will proliferate. Okay. Another is important, that these interactions, when these CD40 and CD40L interact with each other, it also sends a signal to the dendritic cells to produce a very important thing, that this is the third actually event that actually totally activate a T cells, naive T cells. That's called, when this CD40 and 40L ligands attach with each other, they send a signal to the dendritic cells to produce cytokine. So they now starts to produce cytokine. This is the third signaling. So production of cytokines is the third signaling. So I can write here that cytokine. So this is called two signal hypothesis. The first signaling coming from MHC and T cell receptor, and the second is from the co-stimulatory molecule. This is called two signal hypothesis. But third is important for the proper activations of these T cells. This is called cytokine. They secrete, these dendritic cells secrete different cytokines, and they are received by cytokine receptor that are present in the T cells. These cells actually receive the cytokine. These T helper cells actually get activated. These naive cells get activated. So this cytotoxic T cells gets activated by this two signaling process, and also the CD25 interaction with the IL-2, it gets proliferated. So they activated, becomes effector, and the cytotoxic T cells, when it becomes effector cells, called CTL, cytotoxic T lymphocyte. Okay, and when these T helper cells, naive T cells get activated, it becomes effector cells. There are many different subset of T helper cells, and they can be TH1, TH2, TH17, T follicular helper cell, I-Treg, depending on the, so this one naive T cells can become TH1, TH2, TH17 T follicular cells and I-Treg, and there are different subtypes also present in the TH cells, TH2 cells and I-Treg cells subtype. So they can become different effector cells. The reason is cytokine. In my next class I will explain, because this is a different topic, because up to this is the activations of naive T cells. Now these cells become effector cells. You can see these cells become effector cells. Now the cells can activate and get also activated by macrophage and dendritic cells. So in my next class I will talk about how T cells get differentiated and the effector function of these all subtype of cells. Okay, so this is all about the activations, T cell activations.